I will make a start. So I have three areas which I'm going to talk about, which are all interlinked, and so they should flow on one after the other. I'll start by talking about new vaccines and the challenges of developing new vaccines for the remaining infectious diseases for which there are currently no optimal vaccines. And obviously one of the problems with that is related to the different needs of different age groups. And so that will lead on to discussion about the need for life course immunization. And finally, I'm going to move on to some data about novel coronavirus vaccines. So I'm sure everyone is aware that the vaccines have been one of the most important medical advances in our history. And this is just a schematic published by Public Health England, which lays out the development and rollout of vaccines since they were really sort of developed. So you can see from the start that when Edward Jenner first developed the first smallpox vaccine back at the end of the 1700s, and Louis Pasteur developed live attenuated rabies vaccine in the 1800s, that there's been a real acceleration in the development and uptake of vaccines through the middle and later half of the 20th century. And this has continued into the 20th century with some new vaccines and refinements and improvements in many others. So obviously the major success stories in vaccines have been the eradication and near eradication of some infectious diseases, which previously have been major public health problems throughout the world. So the key example is smallpox, which was eradicated from North America and Europe in the 1950s and finally eradicated entirely in 1980. And more recently, polio eradication, which you can see from this schematic back in the late 80s, polio was still endemic across wide areas of the world, but currently it's very much geographically restricted, and we're on course to global eradication. So obviously there have been a lot of successes, but there have also been a number of major infections which still don't have effective vaccines, and the reasons for that are multifactorial, but partly it's because the development of vaccines is a little bit different from the development of treatments, and there are major challenges specifically associated with the development of prophylaxis. So obviously these are products which need to be administered to very large numbers of healthy people and have no immediate benefit to them, but aim to prevent future infection and disease. And so they absolutely need to be safe and well tolerated with a very low side effect profile. They also obviously need to be effective, but effectiveness comes in many forms. So at the very least, vaccines need to protect against disease. And ideally, they should protect against disease for a long time, ideally lifelong. But vaccines should also ideally prevent onward transmission, and that isn't always the same as preventing disease. So preventing onward transmission can be a more difficult goal to achieve. Finally, they need to be practical, and many of our current vaccines are not especially practical. They should be painlessly administered, and needle-free administration methods are not widely available for most vaccines. Ideally, they should be single dose, and many vaccines require multiple doses. And then they need to be easily accessible to everyone across the globe. So that means they need to be stable and easily transported and inexpensive. So all of these are very difficult problems to address. for today, because of my particular interest and for purposes of time, I'm really going to be focusing on the immunology and the effectiveness questions. So we understand that vaccines act by mimicking the effect of infections on the immune system. So following an infection, many infections induce very good high levels of adaptive immunity. So that would mainly be antibodies and T cells, which then protect you against reinfection. And vaccines aim to do that, but also avoid the symptoms and disease associated with the infection itself. So the best understanding we have of vaccine-induced immunity is related to antibodies, which are part of the humoral immune system. And this works by a type of white blood cell called B cells, which recognize pathogens, either bacteria or viruses or other pathogens, and then start to proliferate and differentiate into antibody-secreting cells. And it's these antibodies which, if they reach a high enough level, can bind to the pathogen, neutralize them, and potentially get rid of them before they cause an infection. So this process of proliferation and differentiation mainly occurs in the lymph nodes or in the lymphoid system in humans. And it's in the lymphoid tissue that this process is really optimized, so that ultimately you get these plasma cells which produce a lot of antibody, long-lived plasma cells which last for a long time, and so they are responsible for giving you antibody which lasts potentially for your entire life, and then memory B cells which are there and ready to respond very quickly if you re-encounter the same pathogen or antigen in this case. And memory B cells are really what give rise to this very classic boosting response. So you can see that after the first vaccination, you have a rise in antibodies, but then after a second exposure to the antigen or a booster vaccination, you not only get a quicker response, but in general a much higher response. So the other arm of adaptive immunity that is important is cell-mediated immunity, or T cells. And these are important in a variety of different ways, but partly because they recognize different antigens from B cells. So while antibodies recognize proteins on the surface of the pathogen, T cells recognize proteins which are chopped up within infected cells and then presented back on the surface as peptides. So these internal proteins tend to be much better conserved, and CD4 T cells therefore can help B cells produce more antibody of better quality, and CD8 T cells can directly recognize infected cells and kill them to more quickly clear pathogens. Now, much less is known about how T cells can be harnessed for vaccine-mediated protection. But we do know from several systems that, that T cells are associated with reduction in disease severity. And I've just picked out a couple of key studies from the influenza field. So you can see that in these two studies that prior to infection with influenza, the frequency of T cells against influenza proteins is quite low, but around 7 days after infection, you get this proliferative response, so a vast increase in the number of T cells in the blood. And this is against two different types of influenza, which after a while contract and disappear, but you're left with a slightly higher number of memory T cells. And despite the fact that the frequency of memory T cells at the beginning or prior to infection is quite low, both of these studies have shown that if you have high frequencies of memory T cells in the blood, then your disease severity on subsequent infection is reduced. So in this case, viral shedding is reduced if you have higher frequencies of CD4 T cells, and in this case, total symptoms is reduced if you have higher frequencies of T cells. So I think this is very compelling evidence that T cells can protect against disease severity, although they are unlikely to protect against the actual infection event itself. So going back to why some important infections still have no effective vaccines, I think there are some key globally important diseases which still have either no vaccines or suboptimal vaccines, and these include malaria, HIV, and most of the respiratory viruses. And from my point of view, I think many of the problems we have with developing effective vaccines against these pathogens is due to the fact that these organisms have co-evolved with us and with our ancestors, and developed multiple ways to avoid those immune responses which I've just talked about, and allow themselves to persist in us as their host. So these methods of immune evasion are very complex and multifactorial, but very common ones are strain variation. So if the pathogen develops multiple strains with different antigenic characteristics, it becomes much more difficult to induce a protective immune response that protects against all the different strains. Some pathogens have developed antigenic mimicry, so they mimic the surface antigens of our own cells, and so our own system clearly cannot recognize those. And then other pathogens have developed ways to suppress parts of the immune system and reduce the effectiveness of the immune response. Compounding that is the fact that many of these infections are particularly important in high-risk populations, namely older people and young children. And these populations tend to be difficult to vaccinate, which I'm going to talk about a little bit later. So for the next section, I'm going to highlight some of those different challenges and some success stories and some where we're still working on the problem, using a few example infections. So the first one I'm going to talk about is meningococcal disease, and I'm sure you'll all be familiar with meningococcal septicemia and meningococcal meningitis as major diseases of childhood and young adults. And for most strains of meningococcus, the strategy for developing vaccines has been to grow up the bacteria, to extract the surface capsular polysaccharide, to conjugate that to a protein so that it induces a better immune response, and then this induces antibodies primarily against that particular strain of meningococcus. And that has been successful for the A, C, W, and Y strains of meningococcus. In the UK, you can see that this has, since the introduction of initially the meningococcus C vaccine back at the end of the 1990s, and then later on the introduction of the ACWY vaccine, there's been a steady decrease in the number of cases due to these serogroups. But you can see that this one, the most common, caused by Meningococcus B, has not been affected by that vaccine. And it's been a real challenge developing a Meningococcus B vaccine because the major surface antigen of Meningococcus B is almost identical to a human polysaccharide derived from oxalic acid. And so human beings have tolerance to this antigen. This has now been solved, and this was solved by the application of novel technologies, specifically reverse vaccinology. So in this approach, by understanding and having the genome sequence of the pathogen, it was possible to reverse engineer proteins which are likely to be antigens, and these have been incorporated into this vaccine called 4C-Men-B, or BEC0, which includes 4 different components. And you can see that after priming and then boosting, you get a good antibody response against each of these 4 different components. And this vaccine was licensed in 2013. Interestingly, Meningococcus B is also quite varied in terms of its strains, and you can see from this chart that the match between the strains that have gone into the vaccine and the prevailing strains in different parts of the world is not always complete. But despite that, most studies have shown effectiveness of about 80–85% despite this strain variation. And the Meningococcus B vaccine was introduced into the UK childhood vaccination programme in 2015. So I mentioned strain variation, and strain variation, the classic problem with strain variation is in influenza. And this is a problem that we have not managed to solve yet. Influenza, there are multiple groups of influenza viruses, but the major ones causing human disease are influenza A and B. And these, despite the fact that we have influenza vaccines already, still cause each year 3 to 5 million severe cases and up to 500,000 deaths around the world. As you know, influenza is also a cause of pandemics due to large-scale shifts in the surface antigens, and the most recent one was in 2009. You can see from this dendrogram here on the right that there are 2 main groups of influenza A viruses, but that they separate into multiple strains based on their surface, their major surface antigens, H representing hemagglutinin. So the problem with current generations of vaccines is that they only induce strain-specific immunity, and to protect against the majority of prevailing strains each year, 3 or 4 strains of flu need to be incorporated into the vaccine, and then the viruses mutate very quickly, and therefore the vaccine needs to be reformulated every year to make sure that it matches the circulating strains. And this predictive approach can sometimes not work very well, and on average every 3 to 5 years there is a mismatch in the vaccine with the circulating strains due to an unpredicted mutation or change in the circulating viruses, and so then the vaccine effectiveness falls very drastically from on average about 60%, sometimes down to 10% or less, depending on the strains of virus circulating. So there has been work for many years on developing universal flu vaccines. So the principle of these is that they target antigens which don't change so much either on the surface or inside the flu vaccine, the flu viruses. I mentioned earlier on that T cells are well placed to target these conserved targets, these conserved antigens. But T cell inducing vaccines have been relatively disappointing to date. So there have been multiple clinical trials, for example, with this modified vaccinia ankara based vaccine, which have shown some effect on reduction in the disease caused by flu, but really not met the primary endpoints in those studies. Further research is ongoing, and I don't really have time to talk to you about our research on this, but it may be that the relatively low efficacy is due to the fact that these T cells are not being induced in the right place. And there's a relatively recently described subset of T cells called resident memory T cells, which are sited at the point where you come into contact with the virus, which in this case would be in the nose and in the respiratory tract, and might be better and more quickly getting rid of the virus and conferring more vaccine efficacy. Probably more advanced is the targeting of antibodies for universal flu vaccines. And I mentioned that current flu vaccines really only induce homotypic immunity or strain-specific immunity. And that's because they primarily induce antibodies that target the most variable regions of hemagglutinin. And more recently, it's been discovered that there are conserved parts of the hemagglutinin protein which can potentially be targets for universal influenza vaccination. So this is a study we did back in 2012 following the 2009 influenza pandemic. And on the right, you can see the structure of hemagglutinin, which is comprised, so this is the surface of the virus, and then you have a stalk, which is then attached to a head. And you can imagine that, as well as the protein parts of this head, that there are carbohydrates which stick off the edge, and this is a very big sort of mushroom cloud of a structure. And the top of this protein is hypervariable, so most of it is not required for the function of the protein, which is to allow the virus to fuse with the host cell. But this part, the stem, is critical for the function of this protein. And back in 2009, we isolated B cells and we made monoclonal antibodies and identified several monoclonal antibodies here in red in the schematic, several of which were able to recognize multiple different strains of influenza, some across both H1N1 strains and H3N2 strains. So this really proved that these types of antibodies existed in humans, although they were not common, and could potentially, if preferentially induced, could give you protection against very wide distributions of strains. So this has been taken on by a number of groups which have been trying to construct new immunogens, new antigens, and you can imagine that it's quite difficult to chop off a whole section of a protein and not lose the integrity, structural integrity of the rest. And so multiple mutations need to be introduced to stabilize this conformation. But some of these constructs have been shown in animal models to protect completely against weight loss and completely against death. Still, this is still a very difficult area, and none of those antigens have so far reached a stage where they can be shown to be effective clinically. And but there is a real push now, based on some of those promising data, to develop universal flu vaccines. So a few years ago, the NIH in the US published a target product profile, which is basically this: that a universal flu vaccine should be at least 75% effective, protect against the majority of influenza A viruses, have durable protection for at least 1 year, and be suitable for all age groups. Now, obviously, these are still very difficult targets to achieve. And so in the US, there's been a huge initiative called CIVICS. Many hundreds of millions of dollars are being put into the development of universal flu vaccines from beginning to end, so from research and development all the way to clinical testing. And similarly, the EU and India, through a collaboration, have been funding further research in this area. So I think we're at a stage where we're really attempting to push forward on this problem. But I think that this criterion, being suitable for all age groups, is a particular